Okay, everybody, we're going to begin by going over number 4 on your guided note sheet. So if you would get that out, you may want to pause the video for a second. And I want to go over the answer to the diagram that's at the top of page 3. While you're getting things out, you may also want to get your textbook packet out, because I will also be going over section 5.2, which is about cancer. Okay, so if you'll look at the top of the 3rd page on the guided notes, which would be this diagram here, I want to go over what the answers are. Okay, the letters are a little bit out of order, so I don't want that to confuse you. So we're going to begin with E, which is this cell right here. This cell is an interphase. So where my pointer is, you should write interphase. If we look inside this cell, number 5 is pointing to the DNA, which is in the form of chromatin. So I would like for you to put chromatin with an N for number 5. Number 6 is the cell membrane. Number 7 is the nucleolus. Remember, the nucleolus is where ribosomes are made. And number 8, those are centrioles. The centrioles will be divided during interphase. And remember, the centrioles are only found in animal cells. This next cell here, which is A, is in prophase. We can see it's in prophase because the nucleus is still present and our chromatin has wound into chromosomes, and notice that they're double chromosomes. Each half of this double chromosome is called a chromatid with a D, sister chromatid, and remember this dot is the centromere. However, I feel that number 9 is just pointing to the structure as a whole. So for number 9, I would like for you to put double chromosome. You may also put replicated chromosome. Number 10 is pointing to the nucleus or nuclear membrane, if you want to be very specific. But all the space inside where you see the little dots, that would be the nucleus. Number 1 is the centriole. And then over here in the next cell, which is B, B is in metaphase. Number 2 is pointing to spindle fibers. So remember, during metaphase, the chromosomes line up in the middle. This next cell, which is C, is in anaphase. I know that because the spindle fibers have pulled my double chromosomes apart. And remember, these are now simply referred to as chromosomes, single chromosomes. So number 3, C would be anaphase. Number 3, I would put single chromosome. This last cell down here, you can tell it's splitting apart. Remember, where it splits in an animal cell is called the cleavage furrow. This cell as a whole is in telophase. So in this space right here, put telophase. And down here, what I believe it's referring to is that these are the 2 new daughter cells. 2 daughter cells. I will come back to this handout, so don't put it up yet, okay? What I'm going to do now is talk about cancer. And I think cancer is something that we've all heard of, probably even, unfortunately, have known someone who has suffered or perhaps even died of cancer. Cancer is a very general term. It simply means abnormal cell. People can have cancer in any part of their body, and remember, one type of cancer can turn into another. So if someone, if a lady has breast cancer, for example, if it spreads, then it may later turn into brain cancer liver cancer, people have cancer in their stomach, people have bone cancer, leukemia, which is cancer of your blood and bone marrow. There's just a million different types of cancer. And of course, we do know that it is hard to treat. And that's one of the things that's most challenging about cancer. So I'm just going to go through some of the things that, general things about cancer, just to kind of help you as a whole understand. The information that's in section 5.2 is actually a little more detailed than what you will be tested on. So if there's a few things in there that confuse you, just try to focus on what I say in this video, because I'm going to go over what you really need to know. This is an interesting slide that just answers a few questions that you may have already been thinking about. Is that how often do cells divide? And different cells do divide at different rates. So if you look here, cells in an embryo, so this would be an unborn baby, they can complete cell cycle in less than 20 minutes. And so those cells are dividing, very fast. And just think about how fast development occurs, in organisms. So it only takes 9 months for a baby to form inside its mother's body, especially if you're looking at a human. And that's really not a long period of time when you think about all the things that have developed, in that short period of time. Skin cells, they divide frequently. They complete cell cycle in, even 12 to 24 hours. Liver cells rarely divide. So if someone has significant liver damage, they may have to have a liver transplant because their liver is not going to fix itself, maybe like skin or something else would. And then nerve cells, they don't divide at all. They stay permanently in this phase that's called G0, which we haven't mentioned before. We talked about G1S and G2, but nerve cells don't even get in G1. So if someone has nerve damage, so maybe they have a car wreck and they sever or cut their spinal cord in half, it's not going to grow back together. People that have brain damage, and even sometimes children who get really high fever, they end up with brain damage, and it's permanent. It's something that is not going to fix itself over time, where other parts of our body can fix themselves. So that's why it's always important for us to protect our brain, and why we ask you to wear helmets when you ride bicycles and motorcycles, and to drive carefully, and think of all those things, because if you hurt your nervous system, it's not going to grow back. But back to cell cycle and cancer. Cancer is caused when cell cycle goes all wrong, when cell cycle basically totally messes up. Cells are going to undergo these things called checkpoints. And, if you've ever been on a military base before, you may have noticed that they have checkpoints where you have to show your identification to get past a certain point. Our school building has checkpoints, so, people from the street cannot come in, our school doors unless they have, the badge or the special key card that's going to let them in. So this checkpoint, when you get to it, the cell is either stopped from going forward, or it's allowed to go forward. And so there's several different ones that we're going to look at. Now, there are two that are irreversible. So once you get to them, the cell cycle is going to take place. So if you pass them, in other words. And so remember, this is just kind of reviewing what we've talked about before. So here are single chromosomes, here are double chromosomes. When they're doubles, we're referring to each half as a chromatid. And then, of course, eventually throughout cell cycle, we will separate those again. So checkpoints, again, are stops and goes. So it's just, if you don't meet the checkpoint, you can't go further. If you do meet the checkpoint, then you can get past it. So here are the three major checkpoints. And these are also in your textbook pages. They are described for you on page 118 if you want to look there. So there is a G1 checkpoint, an M checkpoint, and a G2 checkpoint. And so the first one just says, can DNA synthesis begin? So it means, is the cell the right size? Does the cell have enough energy to divide? Are the growth factors present? Is the DNA damaged or mutated? Because if you don't want to make more DNA from DNA that's already messed up. If the cell gets past that checkpoint, it moves to the next one, which is the G2 checkpoint. And this one simply says, is the cell the right size? Is the DNA damaged? Was the DNA replicated completely during the S phase? So we're just checking here to see if S was done completely. Because remember, G1 occurs, then S, then G2. So this is the second one. If the cell gets through this one, it's committed. So like mitosis is going to happen if you get through this checkpoint. The last checkpoint is the spindle checkpoint, or in yours it's called the M checkpoint, which there are only 3. So if you're looking at page 118 with me, you'll see that they're saying the same thing. This checkpoint is asking or looking for if the chromosomes are attached to the spindle fibers, and they're looking for that during metaphase. So what we're trying to see is the DNA going to be divided up correctly? This first checkpoint is the most critical, and if the cell gets the go signal here, it's going to divide. So it's really important for this checkpoint to work correctly. So if you have an abnormal cell, which you may or may not realize this, but our body makes abnormal cells all the time. And what normally happens to them is that they're never allowed to divide, and our immune system just gobbles them up, basically. Just goes and eats them up, phagocytizes them, kills them, basically. But if you have an abnormal cell and it is allowed to pass this checkpoint, then you can end up making a lot more abnormal cells, which is what happens when people have cancer. So this checkpoint is super duper important because this is looking at the DNA of this cell. And if this cell's DNA is messed up, this cell does not need to divide at all. Okay? So if the cell does not get this signal, it's going to switch over to G0 and it's not going to divide, which is good because we don't want it to divide and we want the immune system to go and kill this abnormal cell. If we pass this checkpoint, then we get to go to the next one. Okay? If we don't pass the checkpoint, then we end up here, which is G0. So this is a non-dividing phase. And we said that liver and nerve cells are pretty much going to stay in G0 because they rarely, if ever, divide. All of this is controlled by chemical signals. And you obviously are realizing there's way more to it than I'm telling you, and it's all kinds of intense, complicated stuff going on here. But for what you need to know right now, you just understand that these are chemical signals that are allowing us to say yes or no at these checkpoints. And these go-ahead signals, they're the same thing. They're just proteins that are being made that are giving us the go-ahead past the checkpoint. So this diagram is similar to the one on page 118. Here is your first checkpoint down here. So we're looking for the size of the cell. We're looking for the nutritional state of the cell, and we're also looking for, is the DNA okay? The next checkpoint is up here. And remember, after this checkpoint, we're committed. So we do S phase. So we make more DNA here. And then we get to this checkpoint here at G2. And again, we're just analyzing what we have. If the cell happens to fail this checkpoint, cell division will stop, which is good, which is what we want to happen. That's why we have multiple checkpoints. If you have cancer, these checkpoints won't stop the cell. They will allow the cell to finish everything out. Okay? So in our body, in mine and your body, if we make abnormal cells. These checkpoints will keep us from getting a cancerous tumor. Okay, does that make sense? But if you have cancer, the disease cancer, these checkpoints are failing you and they're not working. Okay? And these abnormal cells are completing division and they're going to divide over and over and over. And that's why people end up with tumors. But in mine and your body, because we don't have cancer, these checkpoints are working for us. Okay? Cancerous cells divide out of control. Cells they know when to stop dividing. So if you look at this picture, in mine and your body, when cells fill a certain area, they don't divide anymore. So just look at this picture. This is just showing you a disc, a little dish or whatever. And you have these cells, and they're going to divide until they fill the dish up. They're not going to overflow. They're just going to fill it up on the bottom. If we take some out, look what happens. They replace them. Okay? Now, in someone with cancer, this doesn't happen. If someone has cancer, these cells would continue to divide because they're abnormal anyway. And they may overflow this dish. And that's why people end up with these tumors in their body. You know, surgeons have to go out and cut out tumors. And you may have heard of someone having a tumor removed that was the size of a golf ball. Or maybe I've heard of people having tumors removed that was the size of a softball. And it was just this big growth of cells that wasn't supposed to be there. Those cells were abnormal, okay? They weren't functioning like the ones here. There are genes that cause cancer, okay? And cancer-causing genes have a name. They're called oncogenes, okay? Oncogenes come from these proto-oncogenes. And if these genes are switched on, they can cause cancer. So people are born with these proto-oncogenes that can turn into cancer later in life. So, take breast cancer. It runs in families. And baby girls are born with the breast cancer gene. And they have it all throughout their life. And it is turned off. And while it's off, it's called a proto-oncogene. But if something happens and it gets turned on, and that's the mystery, we don't know everything that turns these genes on. We know some things, but we don't know everything. Then they can lead to cancer. We also have genes in our body that suppress tumors. Suppress means stop them. Now, that's what we have here. So there are genes that are suppressing more cells from forming. And that's good. We want them to work. If someone has cancer, these genes are not working. Okay? So in someone that has cancer, so someone that has breast cancer, for example, or brain cancer, these genes are not working and they're turned off. They're supposed to keep tumors from forming, but if someone has a tumor, they're not working. And so cancer is a failure of controlling cell division. And if you have cancer, your cells are dividing out of control. Out of control. It's like they've lost their mind and we're making way too many and they're not the right kind of cells. They're all messed up. And of course, what they're doing is replacing the good cells. And that's what makes people sick, is that the cancer cells will outcompete the good cells, the normal cells. And so that's why people with cancer take drugs to kill these cancer cells. And these cancer cells what we're doing is messing up their cell cycle with the drug. Okay? And so when someone develops cancer, it's usually after this has happened. You have cells that are growing that are not supposed to. Cells are not utilizing those checkpoints. Somehow they're escaping apoptosis, which is one of your vocabulary words. That's programmed cell death, which is basically, you see, it's a suicide gene. The cell knows when it's time for it to die. Whatever reason, these cancer cells, they don't know it's time to die. Cancer cells divide at an unlimited rate. Since you have more cells, more blood vessels go to those cells, and it causes them to grow because your body doesn't know, and your body's trying to take care of cells. Well, your body makes extra blood, and that's why they keep on going and going and going. And then they don't stop when they're supposed to. So just like up here in the dish, you see how it's overflowing because of that. And so this is kind of showing you a tumor growing in breast tissue. So here's the tumor here. You can see it's growing. Cancer can be caused by all of these things up here, okay? And these are called carcinogens. And you see, and cigarette smoke literally has been linked to every single type of cancer that exists. Of course, you've probably heard of UV radiation from the sun, radiation, just from chemicals and things like that, your age, all kinds of things, genetics. If you get a tumor, and if it's in an area in your upper body, it's probably going to be close to this lymph system. The lymphatic system goes all over your body. If someone's cancerous cells get into their lymph system, then it can metastasize somewhere else. And that means spread. And I know you've heard probably your parents, grandparents, friends, even maybe people at church talking about this, how someone's cancer has spread to a different part of their body. Well, this is how it does it, through their lymph system. You have lymph nodes all over your body because they make white blood cells. But this is a system that goes everywhere. And so if the cancer cells get into these vessels, it can go anywhere. And that's what's so scary about cancer. Someone can have a tumor in their breast, and the surgeon can go in and even just remove the whole breast or remove the tumor. But if just a few of those cells have already gotten over here in the lymph system, even if the tumor is long gone, that cancer can come back. And so that's why people that have battled cancer, they have to go back and be checked again and again and again to make sure that it hasn't come back. And it may never come back, and it may come back, years later. Tumors come in two forms, benign and malignant. Benign is an okay tumor, okay? It means it's not cancerous. It's not going to hurt you. Freckles are benign tumors. Malignant tumors are the ones that spread. So malignant is bad. It means you have cancer. If the tumor is benign, it means you don't. So if a doctor removes something from somebody, they'll send it off to be tested. And if the results say it's benign, that's good. They don't have cancer. If it's malignant, they do. So then they have to take other steps. So they're going to probably remove the tumor, try to cut it out. They may try to reduce the size of it with radiation. Okay, so all those things are used to help battle cancer. And so this again is just talking about the two main types of treatments, radiation and chemo, which you've probably heard of before. This person is receiving radiation. You have to lie on a table very, very still. They direct the radiation straight at the cancer. Oftentimes, I mean, they go to great measures to make sure you're in the exact same position every time. Chemotherapy, you get it in a drip. So you go in, they hook you up to the IV, you wait, you sit there a couple of hours, the drugs go into your body, and not only do they kill bad cells, they kill good cells. They particularly hate hair follicles, so people lose their hair. They get very sick because the drugs are killing good and bad at the same time. The drugs particularly attack white blood cells, so that means that people on chemo, their immune system is bad and they can get sick from many things. So you never want to go around a cancer patient if you have a cold or if you are sick, because they can get sick really easily. There's a lot of research in cancer treatment, and of course, this is something that people, companies are pouring millions of dollars into to try to come up with better ways to treat cancer. So they're always developing new drugs, new treatments, and things like that. I have an old student that lives in Birmingham, and that's what he does. He researches cancer, and he's trying to, come up with new medications and things like that. So, it's still so much that's unknown about it. If you would, at this point, and this is a long video, if you would turn to page 120 in your textbook packet, I'm going to go over a couple of questions that have been hard for people. I'm going to go over number 1. It asks which of the 3 following terms are most associated with the growth and formation of cancer. And those are going to be oncogenes, which are the cancer genes. The next one is mutation, because normal genes can be mutated to turn into the oncogenes. And your third answer would be metastasis, because that's when cancer spreads. The other question that people have had a lot of trouble with is number 4 and number 5. Number 4 says the p53 gene in mammalian cells is known to stop the cell cycle in response to damaged DNA. If this gene is damaged without repair, what will most likely occur? So it's telling us that this gene stops cell division if the DNA is damaged. Okay? So if this gene is damaged, then what will happen is D, that the damaged DNA will stay damaged, and that cell will divide and a tumor may form. So if that's confusing to you, read it again. It's telling you the gene stops the cell cycle in a cell that has damaged DNA. But if that gene fails, then that bad cell is going to divide. And then the last one is number 5. Patients with type 2 diabetes develop wounds on their feet that do not heal properly. Platelet-derived growth factor, PDGF, is a promising topical treatment for these wounds. Topical means you put it on top. So Neosporin is a topical treatment. Cordaid, hydrocortisone cream, is a topical treatment. So if you put antibiotic cream on a cut, that's topical. Okay. It says, which of the following best explains how PDGF may help in treating diabetic wounds? What you should pick on this one is D, that it binds to the surface of the cells to promote tissue growth and close the wounds. Because it's a topical treatment, it's going to bind to the surface and help the tissue close that wound. Okay? I think you should be okay for the rest. And this concludes our video about cancer. Remember, you need to complete all of your work today before you move on to anything else. Okay? If you have any questions, as always, feel free to send me a message. And I will see you soon.